Hey everyone, welcome back to the channel. If you have spent like more than 5 minutes looking at real genomic data, you would have probably run into a massive headache, which are the gene IDs. So, different databases speak different languages. You might download RNA sequencing data where every gene is labeled with an Ensembl ID, which looks something like this, right? But your biologist colleagues only know the common gene symbol like BRCA2. Okay, so manually searching these on Google one by one is basically impossible if you are handling something like 20,000 genes. Today we are going to build a Python script that acts like a Rosetta Stone, automatically translating hundreds of IDs in seconds, and we will be using the MyGene API. So let's jump into it. So to do this efficiently, we are not going to build a web scraper from scratch. That would take a lot of time. We are going to use an official Python library called MyGene. This library basically talks directly to the MyGene.info database, and we are going to install pandas also to make our output look pretty. So for that, we are going to have to run this code: pip install mygene and pandas. So I already have it installed, so I don't have to install it again. So I will show you just in case you will have to run this code, and after doing that, we will import those statements. So import mygene, and we will also import pandas as PD. Okay, so once that is installed and imported, we need to initialize the MyGene object, which will be our translator tool. So for that, we are going to write this mygene info. Okay, so now for the translation query, let's create a list of dummy ensemble IDs that we want to translate. So for that, we will assign it to ensemble ensembl IDs. Okay, so for that purpose, uh, I have picked a few famous cancer related genes and a couple of random ones as well. So we will copy those. You don't have to know about them right now. We will find it out as we move on. Okay, so there we have it. So we will assign that like this. So to translate them, we use the mg.queryMany function. We pass it in our list, tell it what format our input is in the scopes, what format we want in the fields and the species. So let's start that. We will print a statement first, querying the MyGene database. Right. Results is equal to mg dot what did I say? Query many, right? And then inside that, ensemble IDs scopes is equal to ensemble dot gene. It will be our input format. Uh, After that, fields symbol, then name, and okay. After that, we need to put a comma here. After that, we will assign the species as well. So species is equal to human. Okay. After that, we will return what will be return? Length of results, and we will show how many results have been returned from this query, right? So let's see what happens when we run this. Okay, I think I did a spelling mistake. It should be QUE. Yeah, query many. So it returned four results. So it's when we run this, it basically sends a request over the internet to the database and pulls back the answers instantly. So if we just print the results variable right now, it looks like a messy Python list of dictionary. Let me show you. See, it looks like this, but uh, it is hard to read, right? So since we are moving into data science territory, let's introduce a library you will use every single day, which is pandas. I'm not sure if we have talked about this before in any of the videos, so think of it like an introduction as well. So panda takes messy data and turns it into clean Excel-like tables called data frames. Basically, that is what we use pandas for, to handle data frames, right? So data frame is equal to pd dot uh, data frame. This code will turn results into a pandas data frame. Now what we will do next is df dot drop columns is equal to id. I think there will be an underscore here, and then score. Okay, and now we will display the data frame. Let's try to run this and look at that. We have a beautiful table. The query column is our original Ensembl ID, and now we can see that this ID is basically a BRCA to the breast cancer susceptibility gene. We also grab the official gene name and the enters ID, another common database format, right? So one quick pro tip. What happens if you have a typo in your ID, like, or a gene that was like deleted from the database? So for that, let's see what we can do. Okay, uh, like suppose we take a fake ID is equal to I would write something like ENSG fake 1 2 3 4 5. Okay, let's just take that, and fake result is equal to mg dot query many fake ID scopes is equal to, we will just copy it from here, scopes is equal to ensemble gene. Here we will just take fields is equal to just symbol, and what was next, species is equal to human. Okay, and after that we will convert it into a data frame. Now let's see what happens. You see, the API is actually kind of smart. So what will happen is, let's wait for it, and you can see it simply returns a not found is equal to true flag. Pandas will show this as not a number for the missing fields. So this ensures your script does not crash just because one gene out of 20,000 is outdated. And there you have it. Translating gene IDs is a chore, but with this MyGene library and few lines of Python, you can automate it perfectly. You can easily save that pandas data frame right into an Excel file using this command df dot to CSV, and it will basically save your file into a CSV file so you can share it with your lab. So we used a little bit of pandas today. In the next video, we are going to dive fully into pandas. It is the undisputed king of data manipulation in Python, and knowing it will cut your bioinformatics workflow time in half. So all the code will be in GitHub repository as always. If this saved you from a Google search nightmare, please hit that like button and subscribe. I'll see you in the next video. Thank you.